Hello, and welcome to demola. And today's video we are covering Xylem and Phloem, aka the vascular bundle. So in SNAB, the course that I did, you don't really require much detail regarding this. However, just for the sake of the other exam boards, I will go into a bit more depth, but I will just make it clear where the kind of knowledge for SNAB stops and the other ones continue. So no time to waste, let's get into it.
Okay, so we're going to start with the plant cell wall. And what you need to know about the plant cell wall is it contains cellulose microfibrils. Yeah? So when we say cellulose microfibril, what do we mean? Well, it's going to be a cellulose chain, cellulose being a polysaccharide made of beta-glucose, yeah, which we've drawn as the hexagons here. And we can see there's hydrogen bonds joining in between them, mostly between your hydroxyl groups on different cellulose molecules, or different beta-glucose molecules, per se. Cool. So the hydrogen bonds form between them, and that really forms like a nice solid structure that gives the structural support that the cell is hoping to get from the plant cell wall. Now, we also need to just mention about secondary cell walls. Right? So secondary cell wall forms between the primary cell wall and the cell membrane, and it forms once the cell stops growing. So your cell's done all the growth that it wants to have, then it's formed its primary cell wall, and it's like, okay, let's add another layer of protection by getting the secondary cell wall involved. Yeah, and this has parallel layers of cellulose microfibrils. Again, not really something that comes up too often in biology, but definitely good knowledge to know. And of course, you also have your middle lamella around there as well. Okay, let's get into the point of this lesson. We really want to talk about the plant stem. Cool. And when we're talking about plant stems in biology, we're mainly going to be referring to dicotyledonous stems. Okay, so new term, let's define it. A dicotyledonous stem is a flowering plant which has seeds containing two cotyledons. Yeah, and a cotyledon is it's the embryonic leaves, so it's the first leaves that grow on the plant. Cool. Now, why is it important that we're using a dicot? Well, simplistically, it's literally just for the benefit of just making it easier for us. We do have monocots, however, the monocot doesn't have its vascular bundle arranged as nicely as a dicot, which is why I use dicots in A-level biology. Cool. And this is the structure we're really looking at. So this is a more simplified drawing of it. We have got the other one in the corner, which I'll show you in a second. But when we're talking about a vascular bundle, we're referring to this bit here. So that's your vascular bundle. Now, if we just talk about some general structures around here, we have the epidermis, which is your outer layer. Cool. Now getting onto the vascular bundle, we have three main characters really here. And your main characters are going to be the sclerenchyma, the xylem, and the phloem. Cool. So we're going to talk about them more in detail in later slides, but you need to be able to identify them. So innermost is where you find the xylem. Yeah, it's going to be closer to the middle, the parenchyma. Cool. Then you've got this kind of line here, the cambium. Again, I wouldn't stress too much about the cambium. I don't think it really comes up much, but what you would need to know is, in terms of like dating or aging the plant, they normally look at the rings of a tree, right, or a plant, and your cambium is what forms that ring. So the annual ring, so it forms a new one each year, and that tells you how old the plant is. So annual rings, that's what we use the cambium for. Cool. We also have this one now. This is phloem here. That's phloem. And then on top of that, the kind of captor it all is the sclerenchyma. So know where these things are. It's very important. And sometimes you may be asked to reproduce a diagram of a dicotyledonous stem. So make sure that you are able to kind of draw this out, should the case need to be in exam. Now, what you also might see is this. So especially in SNAB, really, really common question to kind of be asked to identify some things on here. So we're going to practice that together. At the top here, this is your sclerenchyma. So a bit darker, because we'll talk about it in a second, but it has lignin, which is why you get these thicker circles. We have your phloem here. And then this one you'd call your cambium. So often, to be honest, you can maybe get them in the same place, per se. If it was an exam, unless they've really demarcated that it's different, I'd side with phloem, but it's there. And you can see again, you have darker circles again because of the lignin. So this is going to be your xylem. And then on the top here is your epidermis. So identifying this is really important for exams because it does come up quite a lot, especially in SNAB. So make sure that you are. Okay, so we're going to kick it off by talking about xylem vessels. And now, in terms of the function, we'll get into structure in a second, but in terms of function, xylem vessels transport water and mineral ions from the roots to the leaves in a process called transpiration. Cool. Xylem vessels also play an important role in providing mechanical support to the plant. So it like anchors your stems kind of thing. That's what the xylem vessel is really doing. And you can see here we have a diagram of a xylem vessel. Now, things that we need to point out, very important, the xylem contains dead cells. So all of these cells are non-living, dead. Why is that useful? It doesn't have a cytoplasm. Yeah, it doesn't have cytoplasm, doesn't have organelles, which means that you can get a really good flow with the water and the mineral ions through the xylem. Yeah. Another thing, no end walls. So because it doesn't have end walls, again, the water and the mineral ions can just kind of flow very easily through the xylem. Quite a hollow tube as well, so it's got a wide space. It can occupy quite a large volume of your water, and the mineral ions dissolved in it, which is really, really good again. This kind of pattern that we've drawn in here is just to signify that it's lignified. Yeah. So lignin waterproofs and strengthens the wall, so it makes it really, really strong. It also means that because it's waterproof, the water can't kind of pass through the lignified areas, right? And because it can't, it stays inside of the xylem vessel, and that's why it's going to go where we want it to go for transpiration. And then finally, sometimes you have pits. Haven't been drawn here, but your pits are kind of like, we've talked about them before. They're kind of like these thinning areas of cell wall. So say if I have a cell wall here, and it becomes a bit thinner there compared to the rest of it, that place is a pit. So that place would be a pit. And the purpose of a pit literally is just so that water can move between xylem vessels. Yeah? Cool. And you can tell the pits don't have lignin in them, or it wouldn't be able to pass through. Now, like we said, an important function of the xylem is the transpiration stream. And like I said in the intro, there are some bits that are kind of less relevant for SNAB and bits that are more relevant for other examples. So if you're studying AQA or OCR, make sure that you learn about the kind of structure and function of root hair cells, and you also need to know about the apoplast and the symplast pathways for transpiration. So when you say symplast, we mean it's traveling through the cytoplasm. When you say apoplast, it means it's traveling through the cell walls. However, you should look that up because that is more important for your exam board than SNAB. Cool. Now, in terms of transpiration, we're talking about the movement of water from the roots to the leaves, and then it gets evaporated at the leaves so that you begin to get more water. That's the transpiration stream. It gets water through your plant. Now, we also need to talk about mass transport. So this is something that we've defined all the way back in topic 1. But mass transport is the movement of fluid through a tube down a pressure gradient. So very important things that we're saying. We're referring to the movement of fluids, and we're referring to a pressure gradient or down a pressure gradient. Include down a pressure gradient. And the way that they're going to do this is mainly cohesion tension theory. So I've done my best to draw a diagram. But you can see if this is the bottom and this is the top, you can see our water is going to have more at the bottom and less at the top. Yeah, creating your pressure gradient where water wants to move up the xylem against gravity. Now, let's talk about this in more detail in reference to cohesion tension theory. So we have evaporation at the leaves, right? So if we're losing water at the leaves, we create a negative pressure because there's less water molecules there. Now, water is always going to move by mass transport down the pressure gradient, which is against gravity. However, this is going to be pretty hard for the water molecules to do. So what it's going to do is water has this great ability to form hydrogen bonds with each other. And we should have seen this, especially if you're a chemist, then you would have seen this before. But you can see this is a hydrogen bond, the dashed line forming between water molecules. Water can do that very, very readily. Yeah, and it can do it with multiple different water molecules. So you can see that 1 is going to 2. Now, because of that, that is what we call cohesion. Yeah, so because they're cohesive to each other, they kind of pull each other up under tension up the xylem. Yeah, and also another thing that water can do is it can actually form hydrogen bonds with the wall as well. And because it forms hydrogen bonds with the cellulose in the wall, that also allows it to adhere. So cohesion to each other, adhesion to the wall. Now, that helps to get up the xylem. That's kind of how cohesion tension works. So you always create the negative pressure at the top, creates a pressure gradient, and then the water molecules are going to bind to each other and also to the wall to get up. Sclerenchyma is something that we can talk about, but we don't really need to go into too much detail, really. But the main thing with sclerenchyma is that it's very highly lignified, contains dead cells because of the lignin, or the lignin's allowed to be there because of the dead cells. And that means that it has a really, really good structural role. We don't need to know too much about it. It doesn't have anything to do with transport, but it's a very, very good structural role, and it helps keep the integrity of that vascular bundle. Okay, we're also going to talk about Phloem vessels. So Phloem vessels are also a transport vessel, right? And it transports organic compounds or your nutrients, whatever you want to say. It transports them all throughout the cell by the process of, all throughout the plant, by the process of translocation. What is translocation? Translocation is the transport of nutrients from the leaves to the stems, roots, and flowers. Cool. So, in terms of Phloem vessels, we're kind of going to talk about its structure, and then we're going to talk about translocation. So, in terms of its structure, you can see it there. We have living cells, like living cells for active transport. And really, when I say living cells, we have our sieve tube elements and companion cells. So I haven't really shown that very clearly here. But you can see in the middle, this is your sieve tube element, and this is your companion cell. Yeah. So what is really happening is that the sieve tube elements, that's where you have the nutrients flowing through, right? And your sieve tube elements, you can see here, they have they have end plates or sieve plates, and that allows movements of nutrients through. Yeah, so you have sieve plates. You can see as well by the arrows as a two-way flow. But what's important about the sieve tube elements is they do not have organelles. They do not have a nucleus. It's literally kind of just like a cytoplasm. Yeah, and that means that there's no obstruction through the sieve tube element. However, they are living, right? So your companion cells do most of the work. Yeah, your companion cells, they have a bunch of mitochondria to generate ATP. They've got the nucleus. They've got all the organelles you kind of need to make proteins, and they are going to do the majority of the hard work for this whole phloem vessel to function. Yeah, that's the main things in terms of the structure of the phloem, and you can already kind of think in your head about a table comparing phloem to xylem, which is a question that examiners like to ask. So I'm just going to also zoom out so you can see the full definitions. But now we're going to talk about translocation. So translocation happens by what we call a source to sink hypothesis, right? And when we're saying about source to sink hypothesis, what we really want to say, okay, well, first of all, at the source cell, so this might be like your leaves, they've just done photosynthesis, they're going to dunk sucrose. Sucrose is the transport a molecule that we use in plant cells. They're going to dump it into the sieve tube element. Cool. So imagine if we've dumped that into the sieve tube element, what is going to happen to the water potential? It's going to go down, right? Because we have more solute. So your xylem vessel is going to be like, oh shoot, we need to give water to the phloem so that we can have osmosis happening. Yeah. So osmosis happens, water comes in. If we have more water in that space, we increase the hydrostatic pressure. Yeah. So hydrostatic pressure at the source is increased. Now, what did we say? We were always talking about pressure gradients. So if we have a high pressure at the source, higher than at the sink, then that means by mass flow, the phloem contents are going to flow towards the sink. Cool. Now at the sink, we have active unloading. So at the source, active loading of the sucrose into the sieve tube element. At the sink, active unloading of the sucrose into the cell, into the sink cell. Yeah, and this is when the sink cell is going to use it for whatever metabolic function it needs to do. Cool. But we have to think again, okay, we've unloaded a bunch of solutes, which means that our water potential is now going to increase. Yeah, so the water potential in the phloem is higher than in the xylem, meaning the water is going to flow by osmosis into the xylem. Yeah, we reduce the pressure there. So we have a reduced pressure, a reduced hydrostatic pressure at the sink, and increased hydrostatic pressure at the source, which is why you get this mass flow, and that's why you always get the flow from source to sink. So we've got the whole kind of process there. I just wanted to explain it as well as I could before I started just showing you this thing. But I hope that all makes sense in terms of what is happening during translocation. So pause the video if you need to, take some time to read it. We're going to move on to the next section. Okay, we've got some questions for you to try. So the first one is multiple choice, and then you're naming a plant tissue that has lignin in its cell walls. Those are just recall questions. The next you're talking about, you have a cellular cell wall, an electron micrograph, and now you're using the photo and your own knowledge to talk about the structure of a cell wall. So try to do that one. And finally, how the properties of water are related to the transport rule of water in a stem. So you're looking at all of those questions, give them a go, pause the video, and we'll get into it. Okay, so for the first question, we should be happy lignin helps with strength and waterproofing. That was something that was brought up in the explanations, and also just something that you should just generally need to know. Cool. Now, what else? We also have to say, okay, in terms of what plant vessels contain lignin, literally just stick xylem and sclerenchyma. These are just remember them basically. That's all I can say. Just remember it. And whatever you do not mention phloem. They do not contain lignin. Yeah? Cool. The next question, oh my goodness. The next question was kind of talking about, okay, cellulose is in the cell wall. Now, what can you see here? So you need to make reference to something you've seen. Can you see the parallel arrangement? So you should be able to see, you can see a lot of these are running parallel to each other. Of course, there are some in a different layer that are like adding like that, but you can kind of see that. So you're going to want to make reference to that, but really, let's start with basics. What is cellulose? It's a polymer of beta glucose. Yeah, cool. We've also can talk about microfibrils. We said those chains are held together by hydrogen bonds. Cool. Now, what you can say, okay, we saw the crisscross and we also saw the parallel. So mention one of them, yeah? And then what you can also say, if you want to add it on, but you would have got your 3 marks anyway, you can talk about your matrix, you can talk about pectin. These are things that I just like to kind of throw in at the end. Now, the next question was talking about water related to transport rule in the stem. So I should have highlighted, but related to the transport rule of water in the stem. So we're going to want to talk about, okay, when we're talking about water in the stem, we're mainly referring to cohesion tension theory in the Xylem vessels. So you want to talk about cohesion, water together. You want to talk about how they pull each other up the xylem. Yeah. You can also mention about adhesion, putting it to the wall. Capillary action, essentially, I haven't mentioned it in this video. Essentially, it is mass transport by against gravity. This is an OCR paper. That question specifically came from an OCR paper, so maybe it's more important to know for that. And then also, you can talk about, this is a really good point that I liked as well. Although, yes, when we talk about water, we're mainly talking in the context of the xylem. We can also mention it is important for Phloem. Yeah, your sucrose is dissolved in water. That's how it travels through. And also the ions as well in xylem are also dissolved in water. So this is, again, just water as a transport medium. Typically it dissolves things. It's good to know. Okay, let's round this up with the A-star tips. So first things first, be very confident in the structure of cellulose in terms of this is something, especially like polymers and your biological molecules, they're going to come up all throughout your exam. Yeah, so just make sure you're very, very confident in terms of how to draw like a basic glycosidic bond, etc., etc. Cool. Xylem and phloem, although we didn't do a direct comparison table in this video, I think you should just make one. Just make sure that you're very comfortable in terms of comparing the two, because again, it's a question that they can very easily ask in the exam. Also, identification of the vascular bundle on an electron microscope is so important. When I was going through looking for questions, that is literally one of the most common questions that they ask. I know I didn't put it in the video, but it's one of the more common questions that they ask. So make sure you are able to identify it. Make sure you're doing a lot of practice there. And then transpiration, translocation. Although for SNAB, I don't think it's a common question, it definitely does come up in other exam boards. So if you're doing other exam boards, make sure you know that to the T. For SNAB, if you're looking to get a really, really good mark, also make sure that you know it. I think especially cohesion tension is maybe more common for SNAB than translocation, but definitely make sure you can do both. I'd say in general, cohesion tension is probably more commonly asked. Now, linking structures to function is a pet peeve of mine. Just do it. Like, unless they've literally just asked you for structural things, always link the structure to function. Adhesion and cohesion are things that people confuse. So just remember, adhesion is going to be like linked, the water binding to the wall. The cohesion is going to be water binding to each other. Yeah. And then finally, just linking to sustainability. Although again, we haven't covered it too much in this lesson, I think it's just important to know because there's a tensile strength practical. It's one of the core practicals. It's very, very good to be able to link like thinking about the microfibrils and how it makes a really strong arrangement. How can that relate now to that practical? And then also just lignin and cellulose molecules that really provide that structural support. So those are good things to remember to say in your exam. I hope that video has been helpful. If you liked it, please drop a like and subscribe to the channel. Any questions about anything I said or A-level biology in general, please drop them in the comments below. I'll do my best to answer in a timely fashion. We've also got our TikTok and our Instagram, both at demola. Links will be in the description below. But that's all for me for now. So thank you so much for listening, and we'll catch you in the next one. Bye.